became an experts in the biological safety area. We almost became experts without having diploma, and I believe it might be a good sign too, since the problem actually involved everybody. And of course, we need to be prepared in the beginning. So, biological safety, I would not really stop and explain exactly what it is, the definition for you. I believe it's one of the areas of national security. It's under this umbrella and it has lots of factors, and one of it is actually the biological safety. And that's why I would like to more talk about our referral central laboratory of Republic of Kazakhstan. Why have I decided to pick this topic here? Since I believe this topic has this historical value, and I believe it was very relevant. And lately, maybe you have followed this publications in Kazakhstan, maybe area at some publications by CLR, and somehow they are mentioning some of the specialists from Kazakhstan, Russia, some reporters, journalists, in many publications like that. And very recently, I just sat down and counted the number. It was 16 publications altogether regarding the CLR activities. Just imagine how come such big interest is shown to this laboratory. And that is now politics-related topic from the bordering countries, and that's why it become somewhat not justified, and somehow the questions began to ask why. What is the sources of this laboratory? Just not to waste your time here, I would like to just quickly go through my presentation. And this is the building that you see. That is the building of Central Referral Lab that has been built in 2016 and was commissioned in 2016. And since then, in 2017, we've started to utilize this building. So here you have this is the background of the CLR, and so this idea actually started in 2004, not in 2013 or 14, and it was based on the supplementary agreement between United States and Republic of Kazakhstan. Nana Lugar's senator, his name has been mentioned yesterday as well, if you remember, if you recall. And the second person, Nana Lugar, this is the one person, he's the senator. And there was another person, and in United States of America, they have launched the project to decrease the risks of the biological security. And so together with Kazakhstan, they came up with an agreement. So regarding the creation of this CRL in Republic of Kazakhstan that will implement the control over the biological safety. And of course, the main area that was Stepnogorsk, and After this agreement has been signed in 2004, I initially, I would say that from the beginning, I was the witness of the parleys, all sorts of expert conclusions that have been published, and that's why I'm very familiar how the process went. And why have we decided to call CRL? Why referent? That's no question, right? I believe that's understandable now. Central, the idea was that all questions related to Ministry of the Agriculture, the Science, the Ministry of Science, and all of those issues should be under one umbrella, should be controlled by one body, so it will be easier to coordinate the activity. And so initially the discussion took place, and we Kazakhstan experts, and myself included, I was against this idea. We wanted to have, for each ministry, for agriculture, for science, we wanted to have separate referent laboratories for each ministry in different places. So that will be this opportunity. And for that, USA party have stated that it will be too expensive, it will not be integrated enough, and it will not give the coordinated consulate activities, and will not be covering all the biological safety issues. And of course, we had to agree, and in 2011 the construction of this laboratory has begun. Within 5 years, this lab has been commissioned, and here you see the number of the agreement and the more detailed information on the agreement. And here I have some dangerous diseases that are very high spread in Kazakhstan territory. I believe lots of you know about those. There are dangerous and extremely dangerous diseases. Here we have, might be not so clear for you to see, but those are natural foci of plaque in Kazakhstan, just as an basically justification. Here you'll see some natural foci of anthrax in Kazakhstan, just for you to see what is the infectious background in Kazakhstan. Here tularemia, here you see brucellosis, and here you see hemorrhagic fevers, and here you see tick-borne encephalitis. And here I have just shown some information on reference labs in the world. So in the US they altogether have 45 labs. Please mind that it's only related to high risk, extremely dangerous diseases, only for those means. Lots of labs that are situated in USA, they indeed are equipped very well, have highly qualified team. And Europe we have 26 altogether, in Russian Federation 14. For China I don't have enough data. In Japan it's 6. In Asia it's 4 labs. In Arabic countries also they have some of the labs, I'm not sure on the number. And here you see how USA labs are situated here. fully covering the area of USA. Please note how they've been laid out here. And one of the most, I would say, high quality ones in New Jersey, one situated in New Jersey. And here is geography of CRLs in Europe. All of them are reference labs. So here Here you see some of the characteristics of our CRL. Once again, that has been built in 2016. Now it's been 8 years in operations. Some 8,000 square meters, 4 floors. So Ministry of Agriculture is situated there, Ministry of Science, and us as well. And so one of the floor is the common floor to all of us. And here I would say the first one is for our common use. It's a general area, and the rest of the building is being distributed for each entity. Of course, the procurement team is also taken into account for, so it's a wonderful space to work on the highly dangerous diseases. And I think I've mentioned it earlier that everything is that the laboratory is equipped very well, and to build this laboratory the USA have spent 160 million USD. It's almost 80 billion KZT. That was the present from USA to Kazakhstan, and I believe this present to be very nice. And you might conduct some trials, storage, collection, all of that can be done in this lab, but each floor is being under the control of a certain ministry. Once, as I said, one is for Ministry of Health and Healthcare, one is for Ministry of Science, and the other one is for the Ministry of the Agriculture. And here is the missions of our CRL. Here is the mission was, first of all, with the ability to study a wide range of bacteria and viruses on the humans and animals. This remained the same here. Now, the tasks: expanding capacity, consolidation and provision of storage of collection, and of course exchange of international lab practices, and of course for us expanding the capabilities to detect, diagnose and respond to biological threats, and also the consolidation, provision of storage to collection of the especially dangerous pathogens. That was one of the main, and we together, there was an ATAR collection. We've made it clear that it's very important for us to leave it as it is here. At the end, they've agreed. I also need to mention that when we've discussed the idea of the building of this lab, at the time, the head of this commission was Admiral Byrd, very energetic person. And when we've decided to discuss this consolidation issue, I have stated from my side, Kazakhstan is one of the biggest countries in the world, top 9 country, and each region, each country of the region should have its own referral lab. And I've asked for 5 labs, and he said if we will have 5 labs, that will be 2 of a brilliant capacity. And so 1 month later, there was a decision which was on exactly referral lab, level 3 security level. And it was such an unexpected present from USA's side. As of today, it's still operational, and I'm just mentioning some of the backgrounds. And here, integration of the public health and veterinary systems in the Central Research Lab of the Republic of Kazakhstan, that was discussed in the beginning. Each ministry will storage their collection in their places. That was how it was done before. So the storage spaces were not in compliance with all the requirements of the safety. And right now, all the biological safety requirements should be followed for specifically those dangerous pathogens. And this attar collection more or less was stored in accordance with the rules, but the Ministry of Agriculture has their collection hidden here in Astana, and they were to move that collection to our lab. And I will not read my presentation through. So those are functions of CRL and listed here very thoroughly. And as of today, we are implementing easy, but to make sure that specialists are able to work with level 2 free biosafety, biosecurity, that is very important. So the specialists that should be involved in this, they are to go through a certain training program, to be certified, to be trained on a certain methodology, and that is how we ensure the being in compliance with all the requirements. And you understand that any sort of minor deviation from the rules might lead to some serious impact. And here you'll see the structure. And so we have the Ministry of Health involved here. It's Museum of Living Creature here. Now we have the Almaty filial before you. All the labs listed here are operational. Those are well-equipped labs and high-quality employees involved in those operations. Now here is important part to have an understanding, full understanding. So we used to be under different ministries' supervision, and since it was functioning very slowly, we have gathered the joint committees to discuss lots of issues and to come to this one common ground. And there were some collegial decisions and activities that we took from first, second, third floors, as I mentioned, from our lab. And so I believe this joint committee is a good decision for us. Here, just take a look. We have altogether 360 SOPs to ensure biosafety, and other issues such as any operation, any sort of activity should have the standard operation procedure. Without those standardized, unified procedures, we are not making any activities. That is absolutely imperative to have SOPs. So out of that 90 SOPs, each lab might have some specific some specific issues covered in those SOPs. Each might just modify it as well, and so the HSC, our HSC officers, they are strictly supervising the compliance of all of the requirements of SOPs. And here are some of the SOPs that mentioned in the end. We usually approve them in collegial team. That is one common decision that we make. Now here, I believe you're familiar with this topic here. We have all sorts of standards that we are applying. Those have been implemented, those being applied as we speak right now, and each standard being controlled by the committee, by some of the coordinating centers, and almost to the point of having exams to make sure to ensure the compliance with all of the standards related to health and safety, and all of those standards are to be followed mandatorily. Here you see the list of infections that we study separately. Some of them we study together in one team. You see some of the respiratory diseases, some of the anthrax. So we, some of the plaque, that's almost 30 highly dangerous infections of humans, human and animals. And here we mentioning the projects that have been completed so far. As I mentioned, in 2017, in September, we have launched our CLR, and but of course, some of the projects have been commenced earlier than that. So some of them have been completed, some of them still ongoing, and we have lots of projects here. Some of the international projects I have listed out for you that we have completed. Some the journalists and other people are mentioning that we are making some unthinkable activities, and that's why I just want to show you those are very normal, very down-to-earth type of activities, and also very relevant ones within the frame of these projects. And I would say that we have achieved great results. And here you'll see the training, and training is one of the main tasks for us. As I just mentioned, without proper training, the specialists won't be able to work in our labs in parallel from other labs in Kazakhstan. They should go through, specifically management, they should go through the risk-based line assessment training with highly dangerous diseases. That is a very firm mandatory requirement which should not be, we should not deviate at all. Here I'm mentioning some baseline training, and there are some issues that we usually cover with our training process. And after the training course is completed, the certificate should be provided to trainee. I have mentioned already the number of people that went through training for biosafety. within CRL frame. And here we have the third level, biosafety, and I would say that the cycle of the training is being followed very closely. I understand, I see that I'm out of time. Here, I would say that it's not me, it's the clicker that making me late. And this is on our cooperation with other labs, some other centers that we are coordinating our activities with. It's, I would say, very spread list of activities that gives us an opportunity, some international cooperation. Those organizations are very familiar to all of you. And here I'm not gonna just read through the whole text here. All of these projects have been completed so far, all done. This is our standards and so on and so forth. And with that being said, I gave you some short piece of information that this is CRL, and it doesn't have any sort of some sort of questionable issues, nothing like that. It's very down to earth. We are working on a very important issue, the missions, the functions that have been handed over to our CRL, and the activity are being underway. And I believe that the mission that has been laid out very clearly in the agreement between USA and ROK being implemented, and also this lab here has not shown the full capability. I believe that the holding will pay special attention to this lab here, and it will efficiently use the resources and help to resolve many issues. And I believe there are so many questions and issues that will be able to be resolved in this lab. May I please just ask, is that it for the whole information that you would like to report here today? My apologies for being out of time a little. Thank you for your attention. The speaker is not using microphone. Good day, dear colleagues. I'll try to be very brief, so we would make it up on time. And first of all, I would like to congratulate the team of Biotech Center, and of course, I would like to wish you all the luck, success. And let's just move on to my report here. Can you please put my presentation on? So, talking about this biosafety, we of course need to have some tools that will help us to ensure the biosafety. And let's just talk about some prevention means. And I would like to share some results of our activity. So let's talk a little bit on vector capripox viruses, and the reason why we have chosen this virus exactly, since it's a very spread, such a big major virus, 2 DNA, stranded DNA, length of around 150 kbp, and this virus is presented by capripox viruses. So it's the goatpox viruses, lumpy skin diseases virus, and shepox virus. And as any viruses, it is possible to maintain up to 25,000 DNAs. And so it allows us to integrate different types of antigens, and even the whole virus might be involved, and so it's not forming in genome, and it's not oncogenes, and so of course the high therapeutic stability is one of the most important part. So we have decided to go with capripox viruses, and so that might become some unique vector, and it means it usually are the animals, and it's not pathogenic for the people. That's why we've decided to go with this virus again. It's more applicable to ruminant animals. In our institution, we have a very spread knowledge and experience with working with sheeppox virus, and it's been used for a long time now to have the vaccines against the sheeppox virus, and we have obtained the modular virus, and both have been used in our work here. So we have decided to use two types of GWBs, and we've then been creating the nucleotide reductase, and we have created plasmas of integration, which we ensure to implement to the genomes of viruses to see the principle of dominant selection, while we have the dominant of chemical impact. The plasmina that is being in the viruses will remain still. And here you see the schematic of how we created plasmida. We have used the classical scheme here. So we've left right locuses, just an example, and selective gene has been determined, and that is Firmicoli fermentus, that works with that utilizes microphosphoric acid, and we also have created a site. So those are base plasmids for each type, for each species. Now we have schematics of production of recombinant capripox viruses with insertion of the target gene, infections of the lump testicular cells with capripox viruses. So we have recombinant and wild viruses. So on the first stage, we of course used the module gene GFP1, and it allows to follow it in real time to see if recombination have been completed successfully. We have received two viruses. So the green one here in luminous green one, which is the sheeppox viruses and modular dermatitis. And those two viruses have been studied. We have assessed the genes in different types of cells, and we have shown that in transmissive cells the expression level is higher. In non-permissive cells, the virus expresses itself, expresses the protein still. Now we have shown it's 10 stages and still it stayed sustainable. And some detailed the detailed report has been published. You'll be able to see it. And here the viruses became a baseline for our next activities. We worked on scheme of recombination of viral genome.
То есть, используя вирус, который экспрессирует зеленый флуоресцирующий белок, и плазмидную интеграцию со встроенным целевым геном, мы получали популяцию, которая экспрессировала зеленый флуоресцирующий белок, да, вот нестабильная, вот в середине. Так, это как-то можно подсветить. Не пойму, хорошо. Далее, после второго кроссинговера у нас образовывались светящиеся вирусы исходного типа и рекомбинантные, то есть они уже были бесцветными. Это ускоряло отбор, то есть не надо было проводить множественные раунды ПЦР, множественное клонирование, то есть мы вот просто там буквально 2-3 пассажа мы отбирали вот бляшки, которые не светились, и это уже были рекомбинанты с целевым геном. Кроме того, эти вирусы нашли применение, так вот удачно они пригодились при постановке реакции нейтрализации вируса. Если классически мы ставим реакцию нейтрализацию, оцениваем ЦПД, которая проявляется там до 14 суток. Здесь белок начинает экспрессироваться уже через 3 часа после проникновения вируса в клетку. Это тоже было вот оценено. То есть через 3 часа клетка уже светится, если вирус в нее попал. То есть через сутки уже итоговый вот титр вирус-нейтрализующих антител мы можем сказать. Ну, отличается он буквально, может быть, там на 0,25-0,5 единиц, если мы до 14 суток доведем. Но это время, то есть можно эти поправки отработать. Далее, с использованием вот наших акцепторных, так скажем, вирусов, которые экспрессировали зеленый флуоресцирующий белок, мы получили вирусы, ну вот в данном случае 4 вируса, которые экспрессировали генные бруцеллы. Мы показали вот экспрессию на уровне МРНК, это вот слева столбцы, то есть 4 гена.
4 genes, and you'll see in a different type of cultures that we have also proved it by Western blot, and the genes also, we also try to assess how many proteins might be generated within the time frame in cell in 24 hours. We see around 125 to 1 TCD of virus, and so within the cell. So it has been, but it's again, it's using the immune fermented assay, and we have also laid it out very clearly in our report. Here, there are recombinant viruses that have been expressed into Brucella genes. We have assessed in vivo expression and in vitro expression, and so not only in cells, but also in organisms, working on the building of antibodies, and gave some efficiency while being infected brucella, by immunized mouses. So the same formula have been used to develop the coronavirus's vaccine. So we have used the protein of the cell of the coronaviruses and to create the humoral immunity, and in neutralization we have only received some animal testing, so it's pre-clinical testing, and clinical testing has not been conducted at all. So aside of that, we are working on modification of the gene to increase the action to the targeted genes. And since there are so many genes that code in the factors that impact of the immune system of the owner, and so this factor is being deleted by, and there is another direction, that is it's attenuation of viruses that will be shared by my colleague in more details. I, in conclusion, would like to mention that capripox viruses are unique vectors. They have limited host range and not pathogenic for human being. We can use them as a replicative vector vaccines and non-replicative vector vaccines, and recombinant capripox viruses activate both humoral and cellular immune responses. Thank you.
Ну, наверное, если будут вопросы...
If you'll have questions, I'll be happy to answer during coffee breaks, since we have time limits.
Александр Вячеславович, да. Там чуть надо подражать. Да, тут надо.
В смысле? Включите, пожалуйста, микрофон.
Моя лаборатория занимается внедрением технологий, которые имеют прямое отношение к вирусологии.
I would like to mention what is the background of my study. So in Astana, in National Center for Biotechnology, we believe that we are not really dealing with viruses aside of testing or like some type of site testing. So I'm just gonna give you a background of this lab. When we created this lab, the management asked to have this genetic engineering going, full-scale genetic engineering. At the time, the director had it's very applicable, and we are still working on it. And so it's applied sciences, and so the plan was to work on virus genomes, and then once if we have antigens of the viruses, we might work on the immunology. It's not just part of it, it's immunology as a system, as a science. And so we have created different projects, we've got involved. And we have built different gene engineering constructions, and the simple ones and more complicated ones, more complex ones. And as of right now, the viruses have now more applicable can have a practical impact as well. So, for instance, different vectors of the viruses for using the in vitro genes for all sorts of therapies. So different type of vectors of protein that is the autonomously replicating RNA vectors for protein expression in mammalian cells. That is the classical example of the genomes of highly dangerous infections. On them we are basing our vaccines. Right now we're working on a few projects here. That is implementation of Chimerivax technology in Kazakhstan, and that is Chimerivax vaccines against tick-borne encephalitis, autonomously replicating the RNA vectors of the protein expression in mammalian cells. And also, yesterday I have talked about that topic yesterday, and since I have a very short presentation, I'm not going to cover all the activities, but I will talk about the implementation of Chimerivax technology. And right now, the National Biotechnology Center is part of the Biofarm holding. And so I believe that we will be able to implement this project within this holding, and it will be very applicable for vaccination system within the country. Now, we've decided to implement the competences, developing, testing, and registering and implementation of immunobiological drugs against dangerous and especially dangerous infections to prevent biological threats. So, chimeric flaviviruses or Chimerivax technology for vaccine development. And so it actually has different endemic areas, and you see dengue, the yellow fever. I believe like 100 million people have this disease annually. Now, the next one is West Nile, something that is at some point crossing the Kazakhstan border. And so the tick-borne encephalitis and West Nile fever, I would say it's one of the high spread diseases in Kazakhstan, that is for tick-borne encephalitis. In North Kazakhstan, in Almaty, those are areas of high spread situation. And the West Nile fever also being presented here in Kazakhstan, especially it's endogenous viruses, and usually those are the birds who are transmitting these viruses. Now, why we decided to go with Chimerivax? First of all, it's a new technology, and also it has proven its efficiency. And currently, all the vaccines against all the flaviviruses have been built as inoxirion viruses, and the issue is that in the genes is very low, and so in the world you'll need to have in the world the practice is you have your first vaccine, and then annually you need to have a booster vaccine. I'm the participant of this process. I used to live in Siberia for a long time, and in Siberia and eastern part of Russia, the vaccination against tick-borne encephalitis is mandatory. I see that the booster vaccine is being done on annual basis. However, in the world there are some vaccines that have easier way to immunize your organism, and so using this the flaviviruses, they are different because they are recombined based on animal animal viruses, and there is enough to have one dose of vaccine to protect the person from this disease. And within this flaviviruses vaccine, and in the field of immunization against flaviviruses, So it's not against being getting ill with this disease. It's to escape any serious impact of this disease. Now, Sanofi company have developed or not even bought the patents for the technologies to develop this vaccine and gave the Kimirivax name. And they been able to make it on a commercial scale. And there are two vaccines against the flaviviruses, so Imajev and Daglaxa, those two. And in endemic countries, these two vaccines covering more than 100 million people, ensuring high efficiency and high level of safety. One injection is enough to make your body safe. And Kimirivax became very successful since it gave the opportunity to have a vaccine against dengue. That is the first successful case. And based on traditional technology, we never had some commercial type of vaccines. And those are the baseline for technology, how it's being built. Here, modular organization for the flavivirus genome. And here it seems that you can construct them since they are more non-structural. You can select the combination. Here you have different type of planning, molecular planning, traditionally, or you can have So, genome is programming one polyprotein, and it's being broken down to structural and non-structural proteins. And non-structural ones you see C, PRM, and E. And see, this allows to change the genome of flavivirus to get the Hymerian virus. The base will be from one virus to and immunogene-wise, it will be more similar to the used virus. And here what we have is technology for creating flavivirus vaccines using chimerization, Kimirivax, patented by Sanofi. This is the accumulated data worldwide to show that chimerias between different species of flaviviruses are less virulent and then the more attenuant of the parent species. And so what we decided to do within the frame of this holding, we decided to, I believe I need to mention that we're working with the live viruses. It's impossible for us. So we searched for partners, for some Kazakhstani companies and some international companies to discuss how we can deal with the virus. And in Kazakhstan, we won't be able to find a partner as to use the live viruses of encephalitis. We are not talking RNA. We're talking about working and testing on animals. We have found the partners in Moscow, in Institution of Gamaleya, and Irkutsk, the Institution of Epidemiology and Biology. And as of now, with the second, we almost signed the contract. With the first, we are still in discussions. We will try and conduct this works in parallel. It's not because we want that so bad. It's imperative for current regulatory means. And so we will discuss the engineering structure of the viruses, and we will conduct activities here and at their premises. If we won't be able to do it here, then the structure will be handed over to them. It's just something that we will do. And also we will try to, of course, create lone moleculars here. And same time, they will have it done in their premises as well. Since our colleagues have went there a few times already, and so here and in Moscow, and virusological studies being conducted by them. Here in this slide, I believe you see that all the engineering work has been completed. That's how it's done in our national center. And all together we have collected 3 structures that will present the chemirimaxes, and they are only differ from one will express and de-express. You see that is done on purpose, and we also have created the full genome of viruses. And on this slide, I actually forgot to mention that the chemirivax, where we have gene of GFP, it's a bacterial plasmid that we might use, and it has the full implication. And virus gene is used in yellow fever in this stump, and you see the X-ray here from encephalitis. And here you see the results of alive viruses from Moscow. After being infected, it shows you luminous lighting. And virusological results here. Chemirimax being used on the cell level, both. So I would say it's expected results here. It didn't really impact. And here in infected mouses, you see the trends here. So this virus will become immunogenic. And my time is up. I won't talk in too many details. Unfortunately, we're not able to find anything in Kazakhstan that has a live infectious of encephalitis, the stamps. It's impossible to find it here in Kazakhstan. And it's needed to complete the program itself, to conduct the assessment. So here we need to, of course, first infect our animals and then try to make this vaccine. And so Kazakhstan does not have this live viruses, and so let's see, maybe we will be able to do that. So this is the prototype of the Siberian viruses. Here we have try to collect it, and in center we try to work it out here. And so we've been working on, and this activity is done in Moscow. I would say it's very specific information that is shown here, and so maybe it just shown itself in some. And these are different results. I would say, gives you a and so you have the full genome of the viruses here in this case, and those are the name of the partners that we might use. Thank you. Maybe there will be some questions, and since we are not making it on time, let's first have all the questions during coffee break. Let's make it this way. So the next speaker that I will call here, Ekaterina Olegovna, head of the Almaty division of National Center of Biotechnology. Good day. I would like to show you the results of activities that we have conducted against tick-borne borreliosis in the southern and southeast regions of Kazakhstan. So, this is the Lyme borreliosis. The exciters are Borrelia burgdorferi, and the tank is the rodents, birds, deer, exotic ticks, and exotic ticks are vectors. And so we were also to research the tick-borne relapsing fever and relapsing fever. So Lyme borreliosis, you'll see, is clinical manifesting and symptoms. Lyme borreliosis to the tick, the transmission, erythema migrans, rash, system reaction, the facial palsy, multiple erythema. And as of today, we have 22 stamps of Borrelia genera species, and 11 out of them are having a serious impact and harm to the body. We have published 2 reports on collection ticks in southeast part of Kazakhstan. We have gathered 550 species the ticks, and those are 6 types of exodus, and we have seen 12 Lyme borreliosis. So it was a general prevalence. B Burgdorferi was 12%, and prevalence B in Almaty and Zhitosu region accounted for 25%. It's Persulactus ticks, and in Sarkand we also have gathered Persulacatus, and almost half it was showing B Burgdorferi genes, and we have phylogenetic analysis of nucleotide sequences of the FLA-B gene locus revealed B avzei and B garini, and here we've seen Borrelia avzeli that has been detected in Almaty. It's in correlation with European one, and one that has been seen in Talgar. It actually has a separate vector. Now we continued this activity in the with the patients in our region. We had gathered the fever patients, and more or less patients in a good condition under the fever. And as per Ministry of Healthcare, Almaty and Zhitosu regions both are endemic for borreliose, and only 16 Lyme borrelioses have been detected within 3 years, and within 1 year working with patients, we have detected 9 diseased persons, and it means that not enough attention paid for this disease.
Также хочу обратить ваше внимание на то, что мы выявили
Also, I would like to state that seroprevalence has been detected in Shymkent and Taras, and we believe those are transmitted cases from bordering regions. And I would like to just give an information, a case with you. The woman that lives in Jambul region, she here had a 25-year-old woman in a rural area of Jambul region. She presented with fever, nausea, vomiting, weakness, sweating, severe headache, then the arthralgia and malaise. The course of illness was further complicated by encephalitis with symmetric lesions of the midbrain cerebral peduncle and serious meningitis, TBA, and LB co-infection were established by a twofold increase in serum IgG titers between day 21 and day 25 of illness. Both infections responded well to combined therapy with human TBE immunoglobulins and antibiotics and viral drugs and the diuretics. The outcome of the disease was favorable and the patient recovered completely. However, it was not at all the ticks issue, and our expert has been called to the hospital, and our expert have collected the anamnesis. So they detected that she went to Almaty in the beginning of the month, and he just looked at the Lyme borreliose and tick encephalitis, and using exactly what was mentioned before, immunoglobulins and antibiotics as ceftriaxone, we've seen the Lyme borreliosis and encephalitis antigens were increasing, so that actually proved their diagnosis. And that is very interesting because the patient started to recover very quickly, and a year after she didn't have any symptoms of the paralysis, which is not common in these cases. And this type of cases show us how we need to increase the awareness of the population, not only in endemic but also non-endemic regions of the country. And of course, diagnostics should be increased, the level of diagnostics should be increased here. I also need to show you results of the research on the tick returnable fevers group of infections, the bacterial diseases characterized by episodes of recurrent fever that have the periods of epilepsy, which symptoms strong head, muscular and articular pains. It causes death very frequently, and we see at least 20 stamps of the Borrelis. They are being transmitted by firm and non-firm ticks. So we have Ornithodoros 3 types, Rota-Argus 3 types, Exadite 11 types, and Borrelii of recurrent fever haven't been studied in Kazakhstan before. We have collected 942 ticks in south and southeast Kazakhstan, and so we have collected the ticks that have that have beaten the people, animals, and just some free ticks. And so 6% of the ticks had B. borrelios, and Borrelia we have seen in Persulacatus collected Almaty, in Talgar, Almaty, and Sarkan, Shetisu, and Tartakovsky from Shardara in Turkestan region, and Persicus collected from chicken coops at Basinsk and Tulkubas regions in Turkestan region. And we have segmented those, and as we conducted the phylogenetic analysis of nucleotide sequences obtained from the partial sequencing of the A, B, and P66 gene loci, showed that HTBRFB from 24, and we've conducted the multilocus analysis, and so it's 100% similar to what has been described in Japan. And Arcus persulacatus caught in Almaty, we've seen the genes in birds that might harm the businesses related to the that cluster. And so we will continue activities in this field. And since the persiculatus of Miyamoto and Burdorferi has one vector, and they were defined in USA, in Russia. We have analyzed our own, and we've seen that almost 4% of persulatus collected in Almaty here in Kazakhstan were co-infected with Miyamoto and B. Burdorferi. And we first time have seen how spread it is within the population of South Kazakhstan, Almaty and Chetisu regions. We have not determined co-infected patients. However, a very high percent, which is 10%, for infection that is being counted in our hospitals. We are not conducting the diagnosis of this disease, so we need to develop the test system, standard test system, and implement it, and of course increase the awareness of medical staff that we have spread of this disease in our region, so they can pay more attention to this type of diseases. And I would also need to, of course, show my gratitude to all the patients that took place in our research, and to the whole team in Almaty branch, and of course to our colleagues from Epidemiology Center in Moscow, and of course to all the medical staff that has helped us in South Kazakhstan. Thank you for your attention.
Наверное, это вас, наверное, пропустим. Он же просился. Да, вот у нас есть просьба господина Сулейменова, он это ему самолет или куда-то там.
Да, меня перенесли самолет на встречу меня зама.
So Mr. Suleimenov has asked to have a floor since he has a early flight. Please put on, technical team, please put on Suleimenov's presentation. I would, of course, like first of all to welcome all on the behalf of our medical university in Kazakhstan, named after Asfendiarov. So we have the very important subject here, the biosafety. So we have the pharmaceutical school. This biotechnologist is a new specialty, and lots of people from pharmaceutical department choose to go with this discipline here. And looking at different universities, different regions, of course, we see technical universities, and only free in medical university. First of all, we of course looking at the specialists in pharmaceutical area, and to again, of course, for production of pharmaceutical products. So the department has been created 2 years ago, and so first of all, the pharmaceutical biotechnology or safety in the production sites, and so for students that will be specialized in this field, we of course create new lessons, new subjects, and it's farm biotechnology, the principle and methods of obtaining biological products, design and equipment of production of biotechnological drugs, and fundamentals of biological safety in biotechnological production, introduction to the bioengineering, and cooperation with pharmaceutical companies that produce biological products. They have mentioned few requirements, and after coronavirus has hit, Kazakhstan has launched the legislation and related legislation. And also, we launched the pharmaceutical production site, which is in accordance with international requirements. And in Atar, we are participating. It's Atar Biofarm in Karaganda Pharmaceutical Complex, the single one that producing biosimilars, and similar companies, antigen, Kazgil test, and there is, of course, perspective that we'll be having more and more companies that will produce biological products. And regarding this development, I would say, of the pharmaceutical industry, in, for instance, 1993, Kazakhstan only produced 3% of biological products. And during Soviet times, there were, of course, some errors made that this type of production site was situated in Poland, in Hungary, in those countries. And when the USSR has collapsed, it all stayed in those countries. And as of right now, strategically, it's very important to leave it here. In Kazakhstan, we purchasing lots of products from Russia and Ukraine, from post-Soviet Union countries. And farm technology is, of course, to prepare the specialists for production sites. So the system works this way: bachelor degree 4 years, 2 years masters, and PhD is the highest level. We now consider launching masters for biotechnologists, biosafety. We have received such a proposal, and now the dean is considering. And if master's curriculum is being developed, and we might involve all the specialists from this field. We will be working on the curriculum. You'll also be able to become a lecturer. Also, the part of the lessons will take place online. We are in a good relations with OTAR. Our students usually have their internships within 2 years, I would say 10 to 12 activities are now, like the projects are related to biosafety, on how this system works, because there are certain specifics on production of the biomaterials, immunological products, its certain safety regulations, and if also there is antibacterial, antivirus drugs, VIVARs, the utilization is another major part, and of course the continuous training of the personnel.
Так. Или сюда, может. Так, препарат. Что-то в эту сторону пошло. Угу. Ну, говоря о том, что
Mentioning
основная часть производимых препаратов, они, конечно, у нас химические.
usually it's a chemical products that we produce. We usually purchase the substances and biosimilars only being processed in Karaganda, and then it's $100 million USD. Two Russian founders, one is Borombayev, one is Kazakhstani Borombayev. They have the best conditions, and the Karaganda pharmaceutical complex has developed the Sputnik vaccine, and two biosimilars being developed in Turkish company and in Deutsche Farm. And so the lessons learned from the pandemic COVID-19, so Deutsche Farm are not to import their masks, and Turkey has approached us to produce Kazbak, and so the state country, Turkey, that country is very commercially developed. I myself also have been head of international companies from Switzerland, USA, and Pfizer has purchased our company, and I remember when we were purchasing the Pfizer vaccine, this company, we were 164 in line, and so our medical director is from Belgium. He was working on this, and I would say that Pfizer has a brilliant intuition. So there is top 20 in the world. When the Pfizer, at the time when I worked in Pfizer, they were top 10, and in 80s they've seen statin and Diplimar. They have bought all the patents, and the cholesterol problem was then widespread, and then Viagra became very popular. It's been only 10 years and 100 million USD spent, and so they. So erectile dysfunction has been discussed a lot, and so we're talking about male but forgetting about female. So, and because of the impacts of the environment and other problems, so there was problems, and Viagra was reaching the highest popularity. And right now in Kazakhstan, Pfizer wants to localize in Almaty the production site. Pfizer is is proposing a program, the training of our specialists in Russia and other international companies of Pfizer representatives to train, and there are two other international companies that are proposing to build vaccine, immunodiagnosis, medical drugs here in Kazakhstan. Now, is it biological weapon? We still have this question on the COVID-19. So, and we, Krusula Zakaria in the movie said similar to Kazvag that stamp will change, and so we need to work on the files of registration, the rules of the clinical, non-clinical trials, and we are working on pharmaceutical supervision. And those items we work on, and we are training our specialists in our university so they can be part of minor production sites. There is antigen, Otar Biofarm, and Center of Biotechnology is planning to also develop and produce their outcomes and findings. And we shown you here some of the partners. Karaganda Pharmaceutical Complex, 15 best, brightest students have internship in this Karaganda Pharmaceutical Complex. Not just simple internship, they are providing the induction courses, also involving them to the production process, showing all of that. And also Nurlan Mohamed Sadiqtov, the company Antigen Kazvak, when it was developed, the question was where should we produce it? And then also good thing that we have Otar Biofarm, and that has provided this space. And of course we have such a great relation with Research Institute for Biological Safety Problems, and when we used to be students of bio department, we used to work with our own hands, the manual help, and right now they want to do the same, and we still discuss it with our students, explaining how important those specialty is for the QAQC, for the supervision, and we are now cooperating also with Mazgut Altumbayev Center as well. I'm very happy that our students participated two labs here that been built by USA. I see how people getting more and more interested, and then we've been asked, what should we do? This is, I'm saying, this is ATKFK. This is the production site where you see our internship is happening here, Kazjo test. And our students always are very happy in Karaganda. And they said that head of the state had a picture here, and we would like to have a similar picture here too. Thank you, and we're waiting for your proposals for cooperation. We are now working on such a beautiful department with interested specialists. We would like to have multilateral, multi-directional cooperation, and also Dean assumed that the other departments also might have this discipline as biosafety. Biotechnology and biosafety in Gumilev University, there are already two subjects like that. The thing is, in their diploma, they have graduated the pharmacological department and technologist of farm production, and of course there is a list of disciplines of the subjects that they will select. There are some mandatory and some selective ones. So it's only for pharmaceutical department, and for other medical departments it's just as a subject that they might select, just for them to see the biosafety importance and understanding. Thank you.
Спасибо за доклад. Сейчас...
Thank you for your report. Please enjoy a coffee break, and let's gather together at 11:30.
все. То есть я же ездила, там, нет института, я поехала в этот бар, где не надо проехать. Короче, это называется ресторан. Это же как-то... Нет, это уже пишешь, и ты не можешь, короче, я тебе хочу проверить со стороны, личные дела, типа, далеко. Все, да? Ну, сегодня одна так осталась. Просто не будете вчера, просто семья уже страшно.
Есть у нас Юнес Роман Абдуллаевич? Мы немного увлеклись чаепитием, поэтому, пожалуйста. Так. Вот это, наверное, выключить, да?
Что именно? Свет?
Или для этого?
Да не, нормально. Что именно выключить?
Нормально?
Свет? Ну, чуть-чуть мешает.
Если
Ну, так.
вам удобно, конечно.
Так.
Вы в эту сторону направляете?
Здравствуйте, уважаемые коллеги.
Good idea, colleagues.
Хочу поблагодарить организаторов
I would like
за
to
замечательную конференцию
thank
и
our
за возможность выступить сегодня
organizers for such a wonderful event,
здесь.
conference,
Также
and
хочу выразить благодарность
special thank you for
вашему
giving me an
команде.
opportunity to give a speech.
Вы как
I would
раз
like to
помогли
also
нам,
thank our
попасть на эту секцию.
partner company Welt that
Ну,
gave us
многие из
opportunity
вас
to be a
нас
part of this
знают.
section.
Компания GI
I
сегодня
believe
динамичная,
that
очень активно
a lot of
развивающаяся
you
компания.
know us. It's a GI
У нас
company.
очень
We're a
быстро,
dynamically
активно,
growing
постоянно
company,
обновляется всё,
and
выходят
we
новые секвенаторы, новые
dynamically
продукты.
updating
Честно
everything.
говоря, мы не только
Sequencing
обучаемся на
technology.
чём-то,
We
только
not
вот
only
обучились, а потом уже что-то новое выходит, мы просто сами
training, we're
успеваем.
training, and right away we are
Ну, не секрет,
producing the
что
new
компания
product. And it's not a
находится
secret
на,
that the company
скажем, на всех континентах
is
практически, то есть 1000
represented on
сотрудников,
each
с
continent,
огромными
has 1,000
возможностями
employees,
с точки зрения поддержки
and
пользователей, там, с точки зрения
and
аппликационной поддержки, то есть
from the
готов
application
дать
point of
и
view, of the
планирование
maintenance
постоянно,
point of
ну,
view,
и поддержки
library
сервиса
preparation,
отдела,
and service maintenance for the equipment. The client service of the sequencing technology is very good, and the new technology, new platforms that we have here, we are conducting the trials here, the patents, and we have hundreds and hundreds of patents. And just to see the data from that we have obtained from MGI is fantastic, and I believe that everybody is aware of NGS, and it becomes integral part of each lab in different industries. It's very hard to work without NGS, and here I would like to mention that NGR company provides this solution to have a full cycle of NGS from the sampling to the analysis of data, and we have automation units to help to optimize these libraries. Since we're talking about the multiple samples where automation is imperative. Now we also provide the computers, the servers to process the data, and we have everything in our profile. And I would also like to mention few items here for automation here. This portfolio is being updated continuously, and we have some look at specifically at GSTP that is from containers with bloods usually to Oppendorf. So this is the main task of this equipment here. In the left-hand side, MGISTP and NE32 and NE384 for DNA destruction. And DNA it's more of a production, let's say, unit. And also we have some other stations for samples preparation, as MGS P100, and also here have MGS P960 for a high production lab. You will be able to provide a multiple sampling processing, and Smart 8 is last year's innovation, and of course we have MGI FOP has sequencing technology in it, from sampling to sequencing. So that will help you to conduct the whole process and also prepare the library, the report, and only thing that you need to just push the button, and that of course helps you to not waste your time. Here we have some GLab, and it's not of course a full list of our equipment, but GLab is fully automated lab, and GLab is a very complicated equipment. We're still not fully aware of its capacity. It's just I wanted to show how technologically developed we are. And following is the information on sequencing, of this list of equipment, starting from the smallest unit to the biggest. And E25 is the smallest one. The cell is 25 million reads, and capacity, it's a small, fairly small targeted panels. It's a very fairly small unit. You can just simply put it on your desk. G99 is a low pass, it's the roll doors. It's very convenient one, helps you to prepare the cartridges, and it has a unit to wash the cartridges in it. And so who worked with G-series sequencing machines, they know that we have some certain filters that we need to use. Now we have the one that is tell that the oldest one, so many people is being using, because it's one of the first sequencing machines that have been developed by us. and now G50, that was, and G400 is one of the best ones in the market, and here T7 is one of the most productive, highly productive one for cells from one launch, and so it's the biggest sequencing machine. It's one that is huge. Usually is being purchased by bigger labs for the national level ones, and the TOX2. This is what we call it the monster. It's open circuit sequencing. It will occupy a full room, and it allows you to sequence more than 100,000 samples per annum, and it's multi-module major machinery. And also I would like to mention that aside of this station for automation and sequencing, we also have the trials kits for biotech, and they show up all the time. So we have NIPT one and PGT and full genome and single cell and the transcriptomics field as well. And we just might select the most fitable one for you. So lower. So you need to look at your capacity, or how many samples would you plan to sequence. Few words on our technology. And the biggest difference is nanoballs, DNB, you can call them DNA. So from linear object we cycling and we producing a very long line from 3 to 500 copies. And the main strength of this process is that from one matrix you will produce all of these copies. When with PCR you will have duplicates. This is a high risk, and it's the chaotic reactions, and it's just kind of results in numerous duplicates. And now second is the structured chip, specifically designed flow cell with points for fixing DNB, and it's of course mixing your samples to get the cross signal. And another one, the third one, is standard NPS nucleotides with fluorescent labels that detach at each cycle. I would say that is the third component that differs us from the other production companies. And here a little more on the sequencing process itself. And I would say it's standard, it's fairly unified. So we have standard NPS, we call in bases. So this light signal will transform to image, and image will show you the sequencing nucleotides, and so loading DNB, and then your fast TQ, you're receiving the fast TQ. And here a little bit about the preferences on DNBC, and so we that is being worked from one for free 400 copies. And if we're gonna look at all of the strong suits, then of course there is no collection of the errors, and
соответственно, контролируемую полностью
And so
реакцию
it's
Promise
fully
Application.
controlled.
Ну, вот, что все это очень
And
важно
all
при
of that is
секвенировании.
very important during sequencing process.
У меня немного зависает, прошу прощения. Все нормально, да? А теперь я перейду, я расскажу буквально про несколько продуктов. Это полногеномное исследование. То, что у нас одно из наших решений, то здесь все начинается с выделения ДНК магнитными частицами, как всегда. Проба подготовка, она состоит из сейчас 5 этапов. У нас фрагментация и аdenylирование, и восстановление концов. Раньше были 2 отдельных этапа, а сейчас они соединены вместе. То есть происходит параллельно, это один этап стал вместо 2, что значительно сократило подготовку библиотек. То есть полностью подготовить библиотеку теперь можно за 2-3 часа. Раньше было около 5 часов. А так это стандартная фрагментация, она либо механическая, то есть с помощью ультразвука, либо ферментативная, потом восстановление концов, аdenylирование, лигируются адаптеры, делается ПЦР или не делается, если это без ПЦРный протокол, такие у нас тоже есть. Зацикливаются линейные фрагменты, ампликоны и делаем ДНБ и секвенируем. Собственно, вот и весь процесс, я его вкратце упомянул. И здесь представлены наборы, которые у нас есть, вышли недавно как раз. У нас исторически так сложилось, что одиночные баркоды, сейчас мы переходим на двойные. И вот, собственно, 2 колонки справа, это новые наборы, которые двойные как раз, и которые значительно были оптимизированы как раз, чтобы получить данные высокого качества для разных образцов. Ну, есть и один из наборов, это как раз ферментативная фрагментация, а другой механическая. Ну, то есть тут можно подбирать по вашим как раз нужным. А следующее это... Так, я... Тогда просто...
Pathogen fast identification kit. We have this solution, this software solution that allows you to have your samples being analyzed on the number of pathogens and available for 14 host species. And here is the database. Includes approximately 27,000 species, so fungi, virus, bacteria, archaea, protozoa, parasite. So it's just 2-component solution. You will be able to analyze the data. And here, I will just skip all of this here. And in conclusion, I would like to add, for researchers working on in agriculture area, in selection of the cattle, we also have some interesting technical solution. There is a low-pass WGS with low coverage area, and this gives a big opportunity, and it's fairly cheap for the projects where you planning to assay multiple samples. And here is the kit, and thank you. If you have any questions, please ask me during a break. Thank you. If I may, you wonderfully have listed all the preferences of your equipment. And what is the participation level of your company or involvement of your company in development of the functions of those equipment, or do you really making sales? Not clear on your question. So development of this technology, the company itself doesn't develop it. We are the producing. What about the development? I believe he means technology. Who develops technology? Now, the answer is MGI company develops all the technologies that being produced by the MGI company. Thank you.
Следующий доклад.
Next up is to the speech is given to Alexander Borisovich, Mr. Shustov. Good day. Thank you for this opportunity. I would like to thank you for this opportunity to give this speech, and I would like to cover my topic here today. Along the 6 years we've been working on this research, and this is on the dangerous and highly dangerous diseases in Kazakhstan. We worked in conjunction with National Center of Dangerous Infection and also with Veterinary Center of Kazakhstan. And so the biggest strong side is we didn't have the impact from the outside world, and we had scientific support from Jules Verne scientists. We have studied Brucella abortus. Altogether, we've studied 213 stamps of Brucella abortus, starting from 1943 historical data. So we started with MLVA typing, so circling genotypes, and in the beginning we have selected 49 stems, and based on MLVA results, we've seen only 28 genotypes classified, and so the genotypes that have been collected in one area for a short period of time of 3-4 years, they've showed one genotype. So it was sort of like a peak spread, and this clade A and clade B that is circulating in African region in different areas, C1 and C2. C1 is Eastern Eurasia, C2 is West Europe, 7 North and South America. And so we see that 46 stamps from Kazakhstan and clustering in C1 type. This Brucella abortus S19 104M both. And so vaccines might be able to remain in animals for a long time, and usually they discharge via the lymphatics and increase the titers. And so for clade C1 stamps, we see that they've been all formed. And they've been formed together with the stamps that have been mostly, or strains, should we call them, closest to the Russia, Georgia, and China, and also in Italy in some cases. And also you'll be see see that even though the genotyping is similar, it was discharged in different areas in different times. So we see that free cycling of animals is being still available, even though last few years it has been changed, but maybe earlier in 2000s it was in a free flow, and so we need to take control over this process. And you see at least similar nucleotides from different areas, and also from one area you see the circulation of different genotypes. And I would like to say that the technology is not allowing to find the brucellosis in Kazakhstan, so we are not able to differentiate these two types. And so all of the strains being brought to Kazakhstan in the 19th and 20th centuries during the joining the Russian Empire. So this process has maybe kind of assisted this process. So the split in branch D occurred between 1759 and 1842, and the similar has been done for Brucella militensis. We have collected more stamps, and a total of 1675 B militensis. Institution named after Akimbaev have provided those stamps, strains, and we have at least 200 strains, and 156 strains were selected from 9 regions of Kazakhstan, and 8 were isolated from animals, and the remaining 148 from humans. And we see that we just wanted to see those big genotypes comparing to human being from the same area, from the same time period, and how they were able to be preserved during this time period. And there are few lines here of Brucella minitensis. It's America and Western Mediterranean and Eastern Mediterranean, and all the stamps that we had to the subline male EI, the Kazakh strains are grouped in a very restricted part of the B melitensis phylogeny of the Eastern Mediterranean. As you see, all the strains do not have the genetic similarity, and one strain is located separately, and it's possibly a newly introduced genotype. Also, the tourism might have had resulted in it, since this strain most likely being discharged by person. And here, the 88 strains were detected as different line. So 30 strains was close genetically to the Russian and Georgian ones. Also, you might see the Spain, but I believe that Spain has received it from Turkey reproduction. And so, looking at evolution, the Brucella militensis most likely have been originated in the 18th century and later. Before that, we might assume that it was free of Brucella militensis. So for genotyping of phrases, A phrases here, A, B, and C. So A is more popular around the world. So lineage B has been founded preliminary in Northern Asia, Eurasia, in mountainous areas throughout Western Europe, South Africa, and the United States. Now, branch B, C, or lineage C, is extremely rare. Now, genotyping and global distribution. So there are such lineages: Tsiolkovsky, STI, Pasteur, Heroin, TEA011, A0150, A0245. So Tia also has a sub-lineage, and in Kazakhstan we have determined B, two strains, in 2016 in Russia, Stern Amos 6 strains, Volun 2 strains, and Tia Tsiolkovsky, or most cases, Heroin 6 strains, and those infected were the heroin addicts, and so together with supply of heroin from Afghanistan, it was imported here to Kazakhstan. And so here you see STI lineage has 6 sublines, and we see those are endemic for Kazakhstan, which means that STE line was maybe born in Central Asia and then has spread to other countries, and few strains have been determined in Russia at the same time at the moment. Here we would like to see B lineage strains that are not of a character for our area, for our territory, and so it's more identical to Yakutian one and Yamal and Yakutia areas. So as you remember, in 2015-16 we the deers had the Siberian disease, and so one of the proposed ideas was that Batros anclesus might remain for a long time, and so it's important to monitor, and specifically on the mass deadliest disease with wild animals. We will need to conduct a monitor the migration, and also on the routes of the flying lines of the birds. And we also seen Rancella tularensis subhalarsica, and so it's B4 lineage, and it was identified first in Western Europe and USA. Also recently it's been identified in China and here in Kazakhstan. So it means that line B4 are more distributed here in Kazakhstan, in Asia, and B4 lineage, the Asian descends and the further spread in the world. Now, the next we have also determined B12, B42, and so established identifiable genotypes isolated at a distance of up to 1,000 kilometers within an interval of up to 25 years. It may be possible to establish these identical genotypes. So earlier Germany, Sweden has received similar results, So tolerances subholarcita might migrate for a long distance, maintain for a long time. Also, it might be stable for even further time distance. And so now the question is how to control it and how to decrease the peaks. And this data is including the knowledge that the strains came with birds. So see, it has this possibility, the birds. Thank you. I try to make it on time.
Спасибо. Да, есть время, можно задать как минимум один.
We have time for at least one question. Altogether, very nice presentation. When have you conducted this research? We used LMLVA genotype starting from 2012. We collected a good selection, and in 2014 we went to full process, and Brucellus from 1946, and they've been collected in different labs. So we conducted it from 2012 to 2023 within frames of grants, funding of the grants for genotyping. And for yes, DNA institution have provided us such opportunity, and this work has continued. Am I understanding correctly? So we only have one left that is endemical for Kazakhstan and Turkmenistan, and lately we have seen in Russia different, which says a different thing. So we decided to go to Kazakhstan and see its background. For Siberian ones, for the other ones that you have listed here so carefully. No, it's just a partial one. We have only worked with the samples that have been provided to us by a university named after Akimbaev. And we were asking for selection for time periods, certain time periods and certain peaks. And maybe we tried to cover something like around 80. So, but we looking at the peaks. So with each peak, it might kind of discharge 20 strains, and MLVA genotyping, it was something around 120, and Brucellus, not all, because it's such a huge collection in Kazakhstan. I'm not sure if it's been covered fully, but we couldn't. Those are publications. Now, on strains themselves, exciters, well, we do not know the full information, the collection number, identification number only, and that's it. And the passports and the other things like that, we have returned back to the institution and universities, and right now the tendency is to try and preserve this collection, and we'll hope that this data will be used in institutions in their trials. And well, we have checked the collections, and unfortunately the data that you have given us just now, unfortunately it's almost absent. It's not displayed at all. So with veterinarians, I know that they now begun to display it. They actually conduct a similar activity. Okay, it's interesting. Thank you.
Следующий доклад.
Next up, Madame Cappuccini to give a speech Dear colleagues, please allow me personally and on behalf of Algimet Kazakhstan company congratulate our dear friends in National Center here for this anniversary and wish good good luck in the future and more success and more achievements. And let me now proceed with my reporting. Now, my area of responsibility is regarding the resolving issues of the infectious diseases in the poultry. And one of the main issues that is the spread in Newcastle disease in poultry. I believe for a long time we haven't heard regarding this disease. However, on this slide we see that the spread Newcastle disease, according to OIE data, and the number of outbreaks in the poultry worldwide from 2019 to 2024, and data on Kazakhstan, but non-official data in the professional poultry. We know at least 12 to 15 outbreaks in, again, the poultry farming, the professional poultry farming, even though the program of vaccine against Newcastle, so it's actually a few vaccines per annum. The company IDEX that I represent here officially in Kazakhstan within Algimet company frame of work uses this base tetra antibodies IFA. So each IFA is the methodology, I would say, is one of the easiest to implement. It requires less conditions for lab and also gives some sort of tool for doctors to see what is the stress level of your immune system of the body and of course some of the factors. So IDEX has developed this document in front of you to use while using the vaccines from different manufacturers. So the doctors in poultry farms use this forms while conducting IFA, this baseline titers after the vaccination with different type of vaccines from different manufacturers, with the live ones and inactive ones. And so at least some type of range of the titer with the chickens, taking into account the mother immune system coverage. And although we have the monitoring, the constant monitoring in poultries, and even though we have such a high density vaccine system, we still have outbreaks on annual basis, and we know that Kazakhstan is endemic for Newcastle disease, and 18 months ago the outbreaks number has increased dramatically. For a long time we haven't understand why. So numerous poultries have been mentioning the Newcastle diseases. It's different forms of this disease, and usually when conducting when conducting autopsy, always found some issues with stomach. Now we have conducted the risk analysis, so risk factors here. It's not only related to the Newcastle, but also to the H1, the bronchitis, because it's economically very important disease for poultry industry. Also, we have migration or import of eggs from different countries, and so that is the import of hatching eggs and chicks from Russia, Turkey, Uzbekistan, and it's lack capacity of parent flocks in Kazakhstan, since it's very expensive. And we have hatching egg and chicks from the whole world. Now, the number of industrial livestock. Now, Kazakhstan livestock is at least 1.5 million hens, 1.7, 1.2, and long-term use of long-lived birds. So if used to be 420, 450, now they're trying to, certainly with some success, to use it within 720 days. It's all, again, it's all related to parent flocks expensiveness. And another thing is intensity of immunoprophylaxis programs and intensive meat production. Of course, the slaughter, it's 39 to 42 days. It's just what we call conveyor, unstoppable conveyor. So sometimes production sites, they ignore the sanitarian gap that they are supposed to follow. So if boilers are being slaughtered, there should be a sanitarian gap, whether it will be ignored fully or at least will be cut short, and that increases the risk of diseases. And of course, now regarding the vaccines, it's technological violations during vaccination, and there are many diseases, and I just laid out for you the main diseases, and we understand how that hurts the immunity systems for our poultry. And of course, if there will be a new strain, very strong strain, of course the poultry will be very receiving. So in Almaty region, you know, the proximity of poultry farms it's too close. It's specifically Alelia, Agra, Ladaukus. It's all situated too close to each other. It's of course very hard to follow all the safety rules. And now, of course, technological violation during vaccination. If we're talking about with live vaccination, you need to use spray or something like that. Now, in inactive vaccine is usually being given as a shot, and so it means with the latter you need to just catch your bird and then conduct. And of course, these are the costs that being paid. It's additional costs, and of course that will lead to the gaps in vaccines. So some of them will be ignored. And here
Что касается, теперь я вернусь уже непосредственно к самой вспышке болезни
Now let's
Ньюкасла.
talk about Newcastle
Что
disease
произошло?
outbreak.
Вспышка болезни
What has
Ньюкасла произошла
happened?
на промышленной
So
птицефабрике личного направления в ноябре 2023
it
года.
has
Вот здесь на слайде вы
commenced
видите
in November
их программу вакцинации
2023
против
in
болезни
industrial egg
Ньюкасла. Смотрите.
farm. So
То есть у нас получается
here
птица
it
до
means
перевода,
that the
перевод
hens
в промышленное стадо происходит
before
95-100 дней. До
taken to the
перевода на
industrial
промышленное
egg
стадо
farm,
птица получила
it should be
4 живых вакцинаций спрея.
23 days.
Затем
So
на переводе
before
птица
that,
получила
the spray vaccination has been conducted 4 times. Then
И далее
Ulster
каждые 2 месяца в течение
strain
всего продуктивного периода
by
птица
PC,
получала
and every 2
живую
months
азоту
during the entire
спрея.
productive period was
Когда
sought by
появились
spray method.
признаки снижения
And
яйценоскости, слабой
when we
птицы,
see less eggs,
слабой поедаемости, потом начался падеж,
when there was some
который
symptoms
увеличивался
and
по
then
геометрической
the
прогрессии, мы даже и
death rate
не
has
подумали, что
increased,
это
we
может
have
быть
not
Ньюкасл.
thought
Ну, то
that
есть
it should
у нас
it's
же
about
такая
Newcastle disease,
напряженная
because again, as you see,
программа вакцинации. Какой
such
Ньюкасл? Мы
a
понимали,
high
что это некая
density
системная
system
вирусная,
of
то есть
the
этиология данной вспышки это что системный
vaccines
вирус,
here.
но
So,
про Ньюкасл никто
and
и не подумал
no
даже.
one thought about the Newcastle disease.
Ну, однако падеж увеличивался, и на
Unfortunately,
вскрытии потом уже
the death
позднее
rate was
далее мы увидели,
increasing,
конечно, и
and
нервную форму,
autopsy
скручивание головы птиц,
showed
а
all the
затем на вскрытии
results,
мы уже увидели очень яркие
and now we
патологические
had
изменения внутренних органов.
pathological
Как до
change in
20 года.
inner
Ну вот они здесь представлены, собственно
organs.
говоря,
Here you see them.
явные и яркие, вот они изъязвления в кишечнике,
You see the
кровоизлияние в трахее и прям вот характерные кровоизлияния
issues in the stomach.
в
Now
желудке, особенно
the
на вот,
bleeding,
называется, на пояске преджелудка.
bleeding in the
Тогда уже,
neckline.
конечно, мы поняли, что это болезнь
And of
Ньюкасла.
course, we understood right then and there
Из
that it's
научного мира нам очень сильно
Newcastle
помогли, за что им
disease.
огромное спасибо. Это
And
коллеги
scientists
НЦБ
from
и коллеги
Almaty
из
Institution
университета
and
Нисипуль,
National
которые
Center
помогли
of
нам
Biotechnology, and from the university named after Seafood, has helped us tremendously. They gave us they proved that it's indeed the Newcastle disease. We have conducted the histology assay of the tissue, and it was proven once again that it's indeed the Newcastle disease. Here you might see also some of the inflammation of the tissue, of the trachea, and also inflammation of the stomach.
А также
In the
методом
gullet.
ПЦР было тоже
And
установлено, что это именно болезнь Ньюкасла.
and also with another
Коллеги из Алматинского
PC
филиала НЦБ
tested,
помогли нам провести
we have
секвенирование
determined
данного
once
штамма,
again the Newcastle disease.
и после чего было выяснено, то есть
After
почему
which
мы
we
долго
have
задавались вопросом, да, это
determined
Ньюкасла, да, но почему случилась вспышка, несмотря на
Why
вакцинацию.
exactly this outbreak has
После,
happened
уже
even
после
though the
проведения
vaccines were taking
секвенирования
place.
мы поняли, в чем дело.
So we
Это был
understood
новый штамм.
that it was a new
То есть,
strain.
возможно, он проник в Казахстан еще
It
ранее,
might,
когда вспышки начинались только, но мы не знали.
been
Это
imported
штамм
earlier,
генотипа.
but it was
Вот здесь вот
genotype 7
представлена
stem.
филогенетическая древо, по которому седьмого
Now you
генотипа
see
он
the
близок к
the
штамму
DNA
Москва
here,
на 99%,
genotype.
то есть,
It's
скорее
close
всего,
to
проник к нам.
Moskva
Возможно,
stem
завезенными цыплятами,
strain.
завезенным
Maybe
яйцом из России, когда
somehow
мы
it was
очень много везем
imported to us from Russia by the ex-hands. So okay, I see that my time is up. I'll speed up.
Во время вспышки мы проводили
here we see
серологические исследования для того, чтобы дать врачам в
we've
будущем,
conducted
может быть,
the
некий
ELISA
диалог,
studies, this
несмотря на
overseer
вакцинацию, когда
from laying
происходит
hands
болезнь
before
Ньюкасла,
the outbreak.
какую интересную закономерность мы
So
увидели.
this is
Мы
what we've
увидели,
seen.
что при вспышке болезни Ньюкасла у молодой птицы
During the outbreak,
титры резко просели.
the hens, the young
Очень сильно
hens,
понизились. То есть во время рутинного
showed
мониторинга мы
less
видели титры где-то
decreased
примерно
number
на
of titers.
8 тысяч. И
Right.
коэффициент вариации составлял примерно там 50-60%.
So, the using ELISA was something like 8,000, and the percentage of variation coefficient was 50. And these titers indicated post-vaccination immunity against BN and corresponded by high productivity rates in layer. And with old layers, it's almost 30% less. And here in this table we have this comparison of the coefficient within young and old hens. And why is it done?
Собственно говоря, вот выводы представлены на слайде. Мы это делаем для того, чтобы, так как у нас есть единственный инструмент, это серология,
we only have serology monitoring instrument, we need to use it to at least determine something like the breakdown, even though there is a vaccine still. And I believe I'm on time. Thank you.
Если у кого-то интересно, Марина тут у нас рядом, задавайте.
If someone is interested in this topic, Marina is here. Next speaker will be called to the stage, and it's Mr. Zhigailo, PhD Associate Professor, branch of the National Center of Biotechnology in Almaty. So my topic will be study of genetic diversity of coronaviruses circulating in wild and domestic birds population in Kazakhstan. You know that the birds are natural reservoir for many viruses that might infect the animals and also the humans. The most high risk is for birds, and since Kazakhstan is located in the very center of Eurasia, its territory is crossed by several bird migration routes, and so it is important to understand what viruses are circulating here.
Территории очень важны. Вот. Тема настоящего доклада – это коронавирусы птиц.
The topic of today's speech is the coronaviruses for the birds. And so, representatives of two genera of coronaviruses, namely Gamma and Delta coronaviruses, are capable of causing clinical manifestation in birds. Avian infections, bronchitis virus, a member of the genus Gamma coronavirus, is an agent of economically significant infection of poultry. In domestic chicken, IBV causes symptoms such as watery eyes, mucus in the nostrils and trachea, sneezing, choking, wheezing in the trachea, and coughing, depression, weight loss, lethargy and stiffness, decrease of egg production and egg quality, and also other things are observed. And you know that the coronavirus are enveloped viruses and in RNA chain, and they have 6 genotypes, and one of the most popular genotypes is at least 34 different IBV lineages. And there are vaccines available, live vaccines, active vaccines. The problem is that IBV has a low cross-protective layer, and further this lineage of the side vaccine, then the efficiency of immunization is less. So not only it's important to know if circulating within Kazakhstan area or nearby areas, but also it's very important to know on which genetic lineage it's being represented. And before, we never used to have monitoring of coronavirus or within the wild and domestic birds population in Kazakhstan, and we have developed primers for coronavirus monitoring. We have conducted an analysis and also developed the primers to test all the control samples. Here, southeast Kazakhstan, it's the area where we have main migration routes of the birds, and so we had zoo university that has helped us to sample all that area. So we altogether seen 396 birds, and we have positive samples. In wild predator birds that fly during the day, and the other types as well. We have conducted the sequence analysis and phylogenetic analysis of avian coronaviruses, and so we have seen the coronaviruses of two different genetic lineages. Now we decided to look at IBV circulation within the domestic poultry, domestic birds. So Algimed company, big shout out to Marina for collecting major part of the samples in different areas, in south, in central, in in north as well. And so you see the red dots. Red dots are side strains, and a yellow dot is the strains where we conducted the immunization of the birds. And it's again live vaccine, and green ones are the ones free of IBV. And we see that all area of territory of Kazakhstan has this virus circulating. We've conducted genetic analysis, showed that few lineages have been presented in Kazakhstan, and 2 out of them potentially are separate genetic lineage. We need to conduct more research on this, and we will continue. And at the end, I would like to say that the first time in Kazakhstan we conducted the monitoring of coronavirus of wild and domestic birds. We have detected samples, the positive samples in different types of birds. Gamma and delta both have been detected, and they are now in gene banks. And against the domestic birds, we have collected 5 Kazakhstan regions samples, and as a result, we have published our article in the journal. Thank you. I tried to make it short.
Вопросов нет?
Есть?
Is there any questions? Is there any questions? Thank you. Next, we would call Madame Issa Bek, a senior scientific employee of National Center of Biotechnology. Good day, dear colleagues, dear participants. I would like to provide you with my report here for today, and the topic will be the recombinant lumbar virus dermatitis as a vaccine vector. And I would like to, of course, congratulate our National Center of Biotechnology with the anniversary. Wish all the luck. The objective research is virus of capripox viruses, and this capripox viruses has lots of symptoms, and of course it brings negative impact on losing the mass of the cattle, and then it of course decreases the quality of meat production and the abortion, as well as the miscarriages within animals. And as of today, the main methodology is vaccination. In many cases, we'll work with classical methodology with few dosage, but however, standard method with multiple dosage might call for spontaneous mutation. Not only genes, the virulent genes might be impacted, also some of the genes that are accountable for replication might be damaged as well. So it's for the targeted mutagenes, where we distracting some certain genes, the virulent ones, and the aim is to obtain an optimized capripox virus vector that induces a long-term protective immune response against the selected disease. And first of all, to obtain such virus, we have constructed integration plasmids for knockout of immunomodulator genes. So here is the scheme of taping PCR DNA fragments flanking the LSDV genes to be deleted for constructing integration plasmids. Black broken arrows indicated allocation of oligonucleotides primers, and just now Olga have mentioned as well those areas they've been cloned to plasmid by CN-NKPT. Please note here at the schematic, last block here, 66 gene, and see how we try to black broken arrows indicate the location of the position of amplified DNA fragments into the viral genome. You see deleting this gene in 066 gene being damaged by insertion and within sequence that fails the recognition. Now, obtaining a recombinant virus, and it was conducted with immunomodulatory genes expressing a model antigen, and we have used Atrav KZ viruses. It has been discharged in Atarau in 2016. All the discharge has been registered in sequence, and so Antarau KZ has been transmitted to the 008 Delta. When we then came up with Atrau B viruses, where it has the LSDV 06648, and now Pladnida PNDV has been interacted, and we had Atrau BA, and this gave 148 gene. Now it was as a last result we have introduced three triple Atrau 5PGN, and in result we have 3 gene viruses, and it again fails the recognition frame of the gene, and so it's been done with 10 dosage. The usage of PCR has helped us to determine, and again within 10 dosage, and we have determined that this virus is genetically stable. And if you would like to see the results, we have shown the link at the bottom for our presentation. And here we have also determined the safety of recombinant LSDV Atrau 5BGN. We use the cattle, and here we so you can actually see that two calves had some inflammation, which then was diluted very quickly, so it's safe. Now here we also have looked at the immunogenicity and protective activity of LSDV Atrau 5BGN, and we assessed the cattle's. We have selected the calves 6 to 8 months. We have distributed them to 2 groups, the control and the testing group. And in the control group, we injected PBS, and to the trial one, we have injected 6,000 CCD, and we have determined 14 days after the cattle's had the antibodies in level 1, 7.2 logarithms, and on 72nd day 4.4 and 5, and for the same animals 21 days after, the virus has been given to Atrau KZ with 100 TCD, and on the 4th day the testing group animals had fever 41 degree, and they had nostrils mucus, and on the day 7 there were some inflammation on the skin that on the 15th day the necrosis has started, and the details of the results of this trial you would be able to see in the article that we have mentioned in the bottom part. So a recombinant virus with knockout of 4 genes encoding molecular virulence vectors was obtained. We found out that genetically this is a stable virus over 10 passages and replicates efficiently in TY, MDBK, and PC cells. So knockout of 4 genes did not affect virus replication in vitro. And experiments in cattle showed that the virus is safe and has protective properties. And this virus has preserved the properties, and it has four vacant places for antibodies or antigens. So it can be used as a vector for polyvalent associated vaccines. Thank you for your attention. That's it from my side.
Скажите, пожалуйста, в экспериментах с животным вы сами участвовали?
These trials with animals, have you participated yourself? We planned, we assessed the titers of antibodies, but we haven't took a sample, blood samples, but we have assayed the samples.
Вопросы есть?
Any questions? Any questions? If no, let's continue. And the next up, the floor is given to Kanat Tursinov. Please, the floor is yours. National Biotechnological Center. Good day, dear participants, dear guests of today's conference. Let me please present to you the topic my presentation. Here in short, I would like to talk about the Toxoplasma disease. I'm sure that all of you heard of it. This disease, as per different data, out of 30 to 60% of population has this disease. It's a fairly big number of people. Clinically, maybe it's not shown itself. That's why it's a clinically neglected disease. Unfortunately, there are groups of people that are very vulnerable. And so it's widespread zoonotic infection caused by the intracellular parasite, and the people with weakened immune system, or had organ transplantation, or the pregnant woman are in the group risk, and they might have really severe consequences of this disease. The fatality is part of it. Fatal result is one of it, and so it actually infects the brains, the neurosystem, and it's actually in a pregnant woman it is being spread from the mother to the fetus, and so the very serious consequences up to the miscarriage. And so animals also have the abortion and the miscarriages. It's usually not overlooked as a toxoplasma, since we have other factors as well. So toxoplasma is basically affects lots of animals. It's the cattle and the birds, the wild, domestic, and of course the other types of animals. And usually the agents are the cats and cats are spreading the eggs in an active stage, and also usually it maintains in soil, in water, in some groceries as well. Those all are the sources of infection, and usually it's meat products. And of course, it depends on your cultural preferences. If some cultures do not cook well done meat, for instance, in France, the percentage of toxoplasma is higher than in the other countries. Same in Turkey, yeah, they have a lot of cats, and the cats are not owned by anyone, and so there is tendencies like that. Also, the rural and if you're gonna look in comparison, the rural area has a higher percentage of toxoplasma infections. Now, the prevalence in Kazakhstan, it's not enough information, not only for animals, but also for people as well. So we have found only one serological study of a rural population for toxoplasmosis, which was conducted in East Kazakhstan 2009, and so it was showed that in Kazakhstan there were only really old data among sheep, 19%, cows almost 4%, and so we have conducted the in Akhmula and Kustanay regions, the studies, and in the cattle we've seen the low percentage, which is in correspondence with the data that is being provided in global literature. And somehow cattle has immune system to it, and again it depends on region in the country, and so around 8% in Akhmula and Kostanay region. And among sheep, it's fairly high result. It's seropositivity in Zerinda. It's above 50. It's 52% in cows, and so it's just approximate data, and so around 40% with sheep, and not enough data to provide for other types. Now, looking above, of course, the aim of this work was to obtain a strain producing a recombinant chimera protein consisting of combination of three most promising antigens for the diagnosis of toxoplasmosis in cows, sheep, and goats. So lots of proteins that being used to develop serological tests, and we have selected some of them. It's only three. It's SAC1, SAC2, GRA7. SAC that are surface protein and low dominant tachyzoids represented. And on GRA7, it's parasitis 1 and it's dominant. Shown the schematics of plain recombinant protein here, and so transformation, selection, cell growth, and protein production, then cell lysis, affinity chromatography, and then dialysis, SDS speech, mast spectrometry, and then we have conducted the determination of expression of recombinant chemistry protein, and here the main results being shown, but we've continued more. First of all, we've looked at the control agents, the isolate companies. This is a primary data without any processing, and so we just now talking about SAC1, SAC2, GRA7. We have obtained great results, illicit results with commercial control setup, and so depending on what is going to be the content of anti-agent, and for different types of animals, we were setting up different number, and we've seen high correlation and high difference in the optical density. So we will need to optimize this data. And next, there was a COS vaccine. Right now, we just conducting the collection, and of course, we will try to work out this serology on different animals, and that's it. And from my side, if you have any questions.
Пожалуйста, садиться.
Да, конечно.
Thank you for your presentation. It was very interesting indeed. I have a question. So, is it relevant? You already used the commercial kit here. Well, it's imported ones and a native protein, and it's far more complicated, so it's very complicated in this state, and so we decided to use the commercial one and just different animals. And I have a question here. You just mentioned that the Kazakhstan population 60%, 16 you said, 16, okay, that was referring to East Kazakhstan. Who gave you this data? So it's international scientists who received it, and the publication was international, but I believe it was done in conjunction with our Almaty scientists, because I've seen their names. Thank you.
Слово для доклада.
Next up is Madina Abaeva. She is the MNS laboratory of NIU BPB. Good day, dear guests. I would first of all like to congratulate the National Center with anniversary and the pandemic of SARS-CoV-2 in 2019 that has resulted in COVID-19 pandemic disease, and the main so it's the surface of the domino that is not allowing to collect. We have during pandemic 19 we have developed vector vaccine against COVID-19 in a molecular lab of gene engineer. We assessed recombinant the viruses of SARS-CoV-2 influenza virus expressing the recombinant RBD protein, and here we have the DNA marker PC positive control, unmodified NS gene of influenza virus, and recombinant NSRBD gene of the influenza virus flu. And so the viruses have shown infection activity 5.5 and have shown high activity and being genetically sustainable according to assay, and it showed its identical and after 5 passages for NS gene and to determine NS-RBT genetic stability of NS-RBT construct over the 5 consecutive passages. And so we have M is a DNA marker, and we see that it shows the 620 basin to show the sustainability. We conducted again 5 consecutive patches of NSRBD on the furon cells basis. The first was the passage level with 150 BP. The first is modified NSRBP gene in the influenza virus flu PCR, and 2 modified RBP gene in the influenza virus with the second passage level 1100 500 BP. And here we showed that amino acid sequence of the modified protein of the recombinant strain of influenza virus. It shows its stable properties. Thank you for your attention.
Выражать стабильность в 5 пассажах.
How was this sustainability shown in these 5 passages? What is the data? How do you determine this? You showed stable. You said with 5 passages it's stable.
Непонятно?
Yes, I understand your question. However, I'm too nervous.
Ну,
То есть
выражается.
у гена
So the virus has its own property and stability determined by preserving the conditions. Okay, right. You said you're preserving the properties. What type of properties here? Expression and activity reaction. Activity, you said, right. The titer is preserving antigen activity, or maybe the duration of PCD or something else, maybe. The activity of TCD, see, that's what I'm asking exactly.
Так, Карина здесь? Так, пожалуйста, слово представляется для доклада Ивановой Карины Руслан.
Next up is Karina Ivanova with her speech. Good day, dear participants, guests of the conference. I would like to give my report on detection and characterization of the blue tongue virus serotype of 9 genome in equine bloodsucker from southern Kazakhstan. Okay. Horse bloodsuckers are the insects obligate ectoparasites, hematophages, from the family of bloodsuckers, which have a wide range of hosts. Blue tongue is particularly dangerous, transmissible disease of ungulates, characterized by damage to the mucous membranes of the oral and nasal cavities, specifically with tongue disease. swelling of the tongue, swelling of the facial part of the head, damage to the limb, and changes in the skeletal muscles. The country's territory and Kazakhstan itself is considered, especially southeast areas, are comfortable areas for infections as such. And aim of the study is to screen for the blue tongue virus in blood-sucking flies caught in the territory itself in Kazakhstan. Sample collection here has been collected in South Kazakhstan, altogether 104 flies, of which 37 were males, 67 females. So all of them are horse bloodsuckers, and we've conducted the molecular identification, and the insects were homogenized, and total RNA preparations were isolated from the homogenators using trizol and using universal primers to segment 10 of blue tongue PCR. 1 of 4 samples were analyzed. Here you'll see the results, and one of the sample showed positive for blue tongue, and the blast analysis showed the Gini ruler designations. So it's electroanalysis of RNA samples isolated from Hippobosa gene. And so with primers specifically selected for the blue tongue genome, it's 10 genes, genomes, applicons were obtained that cover the entire genome, again a set of 10, and all applicons were sequenced in both directions by the Sanger method, and the assembled genome of the identified strains was deposited to GenBank. Now, blast analysis showed that blue tongue identified in the study is not rheostat virus. All segments were similar in nucleotide sequence to the corresponding dsRNA segment. And you will see all covered segments of this exact virus here, the first, second, third, 2 10th segments. They've been collected, but you'll see the sequence decrease. And here I'm showing some phylogenetic analysis results. So A image shows you segment 10 and segment 2, which is in principle determine type. We've seen that the positive sample is type west of the 9th segment. And the conclusion. This work confirmed the results of previous studies that first time the blue tongue has been determined in blood of the cattle, and South Kazakhstan territory has a higher requirement to conduct further research, and there are good conditions to have a spread outbreak of this virus, and that might be imported from the other countries. And the horse bloodsucker is the diseased inuvulates. And so here we have our publication. Please, if you're interested, please read it. Do you understand what is this decide? It's the catarrhal fever of the sheep. So if two viruses will be in contact. The genome itself that has been collected from 10 segments, even the reconstruction of this genome will build new serotype of these viruses. And the main risk of these viruses is it will be identified in the cattle, and each will need to have its own vaccine against it from one serotype to another serotype. Clinically, what would you say? Clinically, I would say the stem that has been determined, it doesn't it doesn't it rarely has clinical symptoms, and it's more vaccine type of virus. Mucus, nose is mucus, the tongue swallowing, and
Ну, ну, без микрофона. Этот микрофон. У нас осталось...
so the speaker is not using microphone.
Большое спасибо вам всем за выдержку, большое спасибо за участие, большое спасибо за доклады. Доклады очень хорошие, содержательные, глубокие. Ну, конечно, есть тут более маститые ученые, есть молодые, которые только начинающие, поэтому, пожалуйста, берите пример со старших, берите пример у кого-то, потому что совершенству нет предела, поэтому в таких конференциях, когда вы участвуете, всегда будете набирать опыта, и каждым разом она будет совершенствоваться. Поэтому всем желающим, начинающим ученым я желаю, чтобы они достигли своих вершин и стали теми, кем вы желаете. Поэтому я считаю, что наш секция вот это завершает своей работой. Если есть к нам вопросы, претензии к нашему ведению, пожалуйста, скажите, мы попробуем вам ответить. Есть к нам претензии или пожелания, или нет? Большое спасибо. Тогда большое спасибо вам всем. Вы все молодые. Я еще раз пожелаю. Я считаю, что эта конференция свою основную часть завершает. Конференция, я думаю, очень плодотворно, организована, хорошо, предоставлены все возможности выразить свои знания, тем, в разных местах, поэтому это хорошая возможность, в дальнейшем можно участвовать. Большое спасибо еще раз всем организаторам, участникам, всем. Ну, нас, по-моему, ждут обед. Потом, насколько? 14:30 в актовом зале, там большом, там будут подведены итоги и все остальные вопросы, связанные с... Большое спасибо.